Welcome, everyone. Thank you all so much for coming. It's so great to see you all in person. I can feel the energy in the room and just very, very excited to be here. I'm Audrey Gordon, the President and Executive Director of the Progeria Research Foundation.
I'm Scott Burns, Board Chair of the Progeria Research Foundation.
And I'm Leslie Gordon, Sam's mom, and Medical Director of the Progeria Research Foundation.
All right.
Oh, hold on, and I'm Sam's dad.
Sam's aunt. Let's get it all out there. Yeah. Tonight we have a very diverse group, including members of PRF's staff and board, our generous donors, our faithful volunteers, those children and young adults with progeria and their friends and family, and over 100 scientists from 17 countries. Isn't that great? All right. I appreciate the applause. I'm going to read these countries out because it's so impressive. It's amazing. Australia, Brazil, Canada, Egypt, Germany, India, Italy, Japan, Republic of Korea, Portugal, Saudi Arabia, Singapore, South Korea, Spain, Sweden, Turkey, and the U.S. Woo, isn't that amazing? All right. So for all you geography buffs, who came the furthest? I don't know. Anybody want to guess? What do you think? Korea? Australia. All right. Who here is from Australia? Ted, you get a prize.
Oh!
Get a PRF hat. And these are very few and far between, so we'll get that to you in just a minute. Congratulations. Some of you have been supporting PRF since our very first scientific workshop 21 years ago. Others of you have joined us along the way, and some of you are brand new to progeria. We want to thank you all. We thank you and love you all for being on this journey with us to the cure. Oh. Thank you.
Okay, it's my turn. Wow, it's just so great to be back in person. It's kind of overwhelming in a good way. And it's been, 4 years, I think, since we've had an in-person meeting. And we did have a very successful virtual meeting a couple years ago. But we're back together again, and it's pretty awesome. And on behalf of the Board of Directors of the Progeria Research Foundation, I want to welcome you all here to Boston or Cambridge, Massachusetts. Yeah. We do have a board member here, Paula Kelly. Can you please stand up and wave to everybody? Paula Kelly from our board. And I want to thank my sister-in-law, Audrey Gordon, our executive director and president. Amazing, amazing work. Thank you for your commitment for 23 years. Incredible. I'm going to clap for that. Yes, I am. I'm going to clap. Okay. So I'm going to try to emulate my son Sam Burns a little bit, which is really hard to do, and just go off script. He never had a script, as some of you know. Many of you may know when he would get up here. Aren't you going to prepare? I'm good. I'm good. I'll get up there. I'm fine. I just wanted to reflect on actually a lecture that Francis gave 6 months ago during commencement weekend at Brown University, and it was a 45-minute lecture, and I sat there. It was the Frank Rothman lecture. And I sat there listening to him. Before I talk about what I was feeling as I listened to that, I wanted to also recognize Frank. Frank passed away a few weeks ago. Frank has been part of the Progeria Research Foundation, wow, from the beginning, and part of the Medical Research Committee, and a huge supporter of Leslie. And I just wanted, can we just take a brief moment to just think about Frank? Just thanks. Thank you, Frank. So, during Francis's lecture, he was going through the history and everything and all the progress we made, and I was sitting there, and I was thinking about all of you. And some of you who are new, as Audrey alluded to, in this room. And I was thinking about what those 45 minutes summarized was amazing progress. Amazing, right? And I also thought about the hard work, how hard it is, how it must be, because I live with a researcher. You know, those days you go into the lab, and many days maybe, where something fails, right? You got to like get up and start again. And keep moving forward, as Sam used to say, right? And part of his philosophy for a happy life. So I was reflecting on your commitment and my thankfulness to all of you, and to you, Francis, as well, for all of your commitment all of these years, but to all of you, and for those who aren't here who have been committed to progeria research. And I particularly want to give a shout out to those of you who maybe this is your first meeting or it's your first poster presentation. Like, hang in there, right? And thank you for being part of this journey. And speaking of our son Sam, I will just wrap with my brief remarks to say. Oh, I said remarks. That's how Bostonians talk, but I'm from New York, so I could talk about coffee and stuff like that. But so one of Sam's philosophies, by the way, if you haven't seen Sam's TEDx talk, My Philosophy for a Happy Life, please do, because it's amazing, but he also has over 90 million views. He's number 7 now on the TED channel, neck and neck still with Bill Gates' talk on epidemics. So don't click on Bill Gates' talk. Please go to Sam's talk. But Sam also used to say, as part of his philosophies for happy life, and we still talk about it, is to surround yourself with people you want to be around. Here you are. And now I'm going to clap for you all. Thank you for being here. Leslie?
Well, pretty awesome. Oh, I don't know whether to use a mic or not. Well, welcome everybody. Welcome to our 11th International PRF Scientific Workshop, Race Progeria to the Cure. We are in a race. I'd like to start actually by, somebody did that, thanking the fantastic team that put together this workshop. We actually started pre-COVID, working on a 2020 meeting, and we ended up conducting a successful online Zoom workshop in 2020 that you see here. There were lots of people online, but it's not like being in person. But everybody on that team agreed to do it again, to keep going for this full in-person workshop as well. So kind of like double the work, double the time, pretty amazing. The scientific organizers, Vicente Andres, Mark Kieran, please stand and be recognized, and Judy Campisi, who couldn't make it to the meeting. You guys are fantastic. Never hesitated to say yes. Our terrific coordinator, Justine Mastranzi, and the amazing Joanne Morris, who handled all of the myriad logistics. Thank you, guys. Please stand. You're amazing. Oh, yeah, they're working. Yesterday, our volunteer medical research committee members met to review new and exciting grant proposals. Here we all are in the room. It's a tremendous amount of work, and these volunteers are willing to give their time and talent year after year. They help to shape the basic science that's critical to advancing the entire field of progeria and understanding its relationship to aging. And I want to ask all of you to stand as well as we thank you for this beautiful service. Our fearless leader, Brian Toole, Ted Brown, Maria Erickson, Vicente Andres, Chris Harlingberg, Bob Bishop, Monica Kleinman, and Judy Campisi, and Marcia Moses. Thank you all, guys. You're amazing. Now I'd like to introduce our first session. We have a lot of firsts at this year's workshop. It's the first time we're back together in person since COVID, 4 long years. It's the first time we're meeting in person since we achieved our very first drug approval, which we'll talk about a lot tonight. But actually, I think most importantly, it's the very first time that a young adult with progeria is moderating the opening night patient session. amazing. Sammy Basso, a brilliant, insightful, funny, and forward-thinking young scientist of almost 27 years old, will lead this session. This means more to us than we can say, because it means that we as a community are making real progress. More children with progeria than ever before are entering young adulthood. That would not be possible without the patients and families, scientists, clinicians, volunteers, and supporters in this room. Sammy was one of our very first clinical trial patients, as was Megan, who will speak as well, along with the amazing Carlos, who's part of our younger community. They've been taking lonafarnib for a very long time, and they're here today to inspire us to work better, stronger, and faster towards even better treatments and the cure. So please help me in welcoming Sammy, Megan, and Carlos to the stage.
I'm so excited. Thank you, Leslie, for your beautiful words. And hello, everybody. Thank you very much to be here. I'm so happy and quite scared helping to moderate the patient session. But I'm very proud to have the opportunity to tell a little bit of my life, my experience with the progeria journey, and most importantly, I'm very proud to be with my partners in crime, Megan and Carlos. I thought a lot what I could have told you, and it is quite difficult because many would be the things to say, despite I'm quite sure that the words cannot really express what I feel and what I'm feeling right now. As I said before, as Leslie said before, I'm almost 27. I'm a 27 years old person, or I should say an old person of 27 years. And I'm the oldest person with progeria recognized living nowadays. I'm involved in the progeria research journey since many years ago, and not only because I was born with progeria, but also because I wanted to be as more involved as I can. So I'm actually the spokesperson of the Sammy Basso's Italian Association for Progeria and the international ambassador of the PRF, a role that I'm really happy to share and live with Megan, the one of ambassador. But let's start from the beginning. When I was a baby, progeria was not so visible. I was a quite normal baby, so busy on playing with my parents, as you can see in this picture. Here I was with my dad, Amerigo, my hairy Italian dad, Amerigo. Or as you can see here in this picture, here I was with my mom, Laura, and I didn't believe I couldn't have so long hair as she had that time. They are both here today because, you know, Italian parents are just a little bit obsessively close to their son and daughter. I'm joking, of course. When my parents discovered I had progeria, their life ruined against them. That time there was no information, there wasn't research, there wasn't anything. You don't need me to tell you how our life changed when we knew about PRF and what it means for us to meet other families with progeria. In my case, I was too young to be conscious of that, But it was a pivotal step for me to understand that scientific way would be probably my way. Here it was in the first time I went in lab. I was here in the United States, in the NIH, with my friend Lino. And I can remember that day as it was yesterday. So during the years, I understood how to live with progeria as a kid. Then as a teenager, now I'm ready to live my life as an adult. Here you can see how I feel the overwhelming majority of the time. Here is how I really look like most of the time. And this is who I really am, if you ask the friends. During my life, I did a lot of great experiences. I could not say anything to the amazing trip on the Route 66 I did with my parents and with Ricardo, a great friend of mine, and a troupe of National Geographic. I had the opportunity to see the States from the east to the west, walking on the desert or on an enormous skyscraper, visiting the creator of The Simpsons and the producer of Avatar, and more than this, simply staying with people. I had also the opportunity to receive a new identity. In fact, a Navajo chief in the Monument Valley gave me a Navajo name, Chanagai. That means person with a long way to live. Such a good name to be called with. And in general, to be with people is the thing I prefer in my life. Thanks to my foundation, I had the opportunity to meet so many different kinds of people and to do so many different kinds of events for sustaining the research, for doing a correct scientific disclosure, to help in our little way, the PRF. For example, thanks to the involvement in my foundation, I discovered that I really love to play theater. I love to mask and to be someone different sometime, and in the meanwhile to express some parts of me I cannot express normally. With my foundation, I had the opportunity to meet people who let me to participate in different projects. For example, to be involved in a first aid campaign in my country, or to visit the CERN of Geneva, and many, many others. Anyway, despite the scientific disclosure and fundraising are such serious things to do, we never forget to have fun. That is why my friends and I created the Sami Runners team. We are a team of crazy people running marathon and other races for sustaining the research. We want to spread the message that alone we can arrive so far, but together we can arrive there where alone it was impossible. Like for me to run tens of miles. They are my closest friends, they are my tribe, they are the reason why I'm able to face my life smiling. Why am I saying all these things to you? Why am I telling you my story? Not because I think you are interested on. Well, maybe you are, but this is not the reason why I'm doing that. Nor because I'm a self-referencing man, because I'm not. The real reason is just to let you know that a life with progeria can be so full and so worthy. You still know that, it is clear, but I want to say you that from my perspective of patient, you are the reason why I can say I am living now. You are the reason why I'm still here, the reason why I'm still alive. The reason why I'm damning happy to enjoy all the things that life can give me. With your job, fighting every day harder for arriving to a solution, you are giving me and other patients the opportunity to live our lives. That is why I chose to become a scientist, because I want to do the same. In fact, you are not only the reason why I'm still alive, I'm still able to wake up every morning, You are also one of the reasons why I'm happy to be alive. The world can seem a bad place sometimes, but looking to the scientific community working on progeria, I understand it is not. It is such a very beautiful place to live in. In 2 years, there will be the next PRF workshop. After 25 years from the PRF Foundation, after 25 years from the birth of the research on progeria, in each goal of this type, in terms of time, we must ask ourselves if we are going in the right direction. Well, looking to my story, looking to Megan's one, you'll hear it from her, and looking to the energy of Carlos and many other patients, I think that the answer is yes, we are going in the right direction. When I was diagnosed, 14 years was the lifespan given to me. I'm almost 27. That is what it means to have treatments, that is what it means to have research. We were never being so close to a cure than now, and we will not stop until we will have one. We must do that for the new upcoming generation of kids with progeria. We must do that to honor the people with progeria who are no more with us in the physical world, but who are every moment present in our mind, in our heart. And now, let me to spend some words for thanking someone else. My family, firstly, was here. In Italy, we say that the apple doesn't fall far from the apple tree. Without them, I would not be the person who I am. They fought my battles when I was too young for doing that, but they also taught me how to fight them now that I can. I couldn't be more grateful, despite I don't say them as many times as I should. I want to thank Riccardo, Gabriele, and the other Riccardo who are here too. They are not only my closest friends, but they are also close collaborators inside the board of my foundation, loyal mates with which live my life. I don't have biological brothers, but definitely they are the best brothers I could ever hope for. As a great man used to say, we are trying to live our life, never missing a party. I want to thank all the PRF staff for the amazing job they are doing during these years. Many should be the PRF people to thank. Let me to thank one special person in place of everyone else, Audrey Gordon. She's every time ready to work together, to involve me in so many beautiful projects, and to lead the PRF every day ahead. She is also every time ready just for chatting with open heart with me, sharing thoughts, experiences, and values during the daily life. And finally, I want to thank a special family. Without them, all this great involvement and community would not be here today. Leslie and Scott. They are the reason why everything started. They are the fuel through which all this can go ahead. I cannot imagine my life without you two, really. You are like a mother and a father to me. You are the mother and the father of us all. Thank you for being part of my life. With you and Sam, I lived some of the moments I will remember forever as the best of my life. Thank you very much. Thank you. I speak for a too long time, I know. So you will be so happy to hear someone else. Megan, you are so inspiring. I will not say anything about her because you can hear by her voice. So I'm very happy to let the stage to her. Megan, the stage is yours.
Thank you. Hello, everybody. I think Sammy said a lot of wonderful things in his speech. Very well spoken. I'm very torn in my speech because Leslie's mad at me because I didn't plan anything ahead of time and I didn't send any pictures in. On the other hand, Scott says that I should definitely do things on a whim, so that's what I'm choosing to do right now. Do we have the clicker?
Yes.
Thank you. I do have a few pictures. Thank you. So hello, I'm Meg. I am, just for a little introduction, I am 21 years old, almost 22. I am graduating college in December with a major in creative writing and a minor in science. So much of what I do is not scientific, but I like to kind of take science and bring people in it, which is what I'm kind of going to be talking about here for a little bit. I have been the ambassador for PRF for a number of years that I can't name right now because I don't know how long it's been, maybe 7. And that's been wonderful to come to events and be able to talk and see everybody here and just kind of bring the community together. These conferences are great because we get to see all of the people coming here together who do everything for us and help find the cure and help find treatments, which is fantastic. And another really wonderful aspect of PRF that I think goes a little more unsaid and is a little more rare in terms of like different conditions is that we're a very close community of people. There are only about 200 of us in the world, and it seems like that would lead us to be pretty isolated, but PRF has done a wonderful job of bringing us all together and giving us the resources to find each other and find community and aspects of us that are similar to each other. So, how does this work? We go to a camp called the Hole in the Wall Gang Camp, which is a camp for people with different illnesses where they can come and just be who they want to be and be whoever and get taken care of. So many of us in the United States have been able to come, and even from outside of the United States, have been able to come and just be together. And it's a really wonderful community building place. I met some of my best friends there, Lindsay and Kaylee, who you will see several pictures of. These are not in any order, so... this is Carly, who's also one of my lovely friends. Carly. I am hoping maybe to work at this camp at some point, and I just think it's a wonderful place for people to come together and just find people who are like them, which is very important. I don't know how this works. That's me and Lindsay. I met Lindsay at camp, and she is one of my best friends. She's kind of like my little sister because she's 3 years younger than me, and we can talk about pretty much anything together, which is fantastic. And that's me with Callie, who is younger. I forget how old she is, but this was also at camp. That's all the pictures I have, apparently. But
Next talk.
great. I will not spoil that. Let's go back to this lovely picture. Okay, there you go. Yeah, so I just wanted to highlight the aspects of community that PRF has managed to bring together. Even from the very beginning of these trials, PRF has paired children together so that we weren't alone. So even before at least I was really aware of my condition, I was around other people who were similar to me, and that just meant a lot to have that space for me and for the parents and for everybody involved. It was great. So it's lovely to remember that in light of all the science going on, that there's also the people involved, and just, yeah, it's a fantastic, fantastic organization. So thank you, everybody, for being here tonight.
Thank you, Megan. You are so inspiring, as every time. And I'm sure everybody will have absorbed a lot from you. Do you? Yes. Great. So, I think it is now time to give the stage to the younger of us, Carlos, but I think we have a video for presenting you, right?
Yes, we do. And I think to start my speech, it's not in spiral or like the other ones you two have. I didn't plan nothing, and I didn't send pictures. I have just a video because I make little funny videos for Instagram, so I have a video to show.
Great.
É só para avisar que semana que vem eu entro em férias, tá bem? Ok. Como é que eu posso entrar em contato com você? Espera eu voltar de férias. Ah, mas e se precisar resolver alguma coisa importante? Em caso você pode ligar para outra pessoa, né? E se for uma emergência? O 90?
Well, yeah, thank you. So I'm just gonna have a little story about me. I am 12 years old, the youngest one, still in 7th grade, so I don't have any job yet that I still want to do. But I would say chemistry and cooking interest me. Let's see what that leads me to. And I lived in Brazil for a long time, and when my parents also discovered progeria on me, it was a big difference for us because progeria is just here, and we went to a lot of doctors, and each one told a different story, which was kind of concerning. And they wanted to do tests on me, so we went to a specialist in rare diseases on a big city state of Brazil to see if he knew. And that's a story that makes my mom very emotional when she tells others, because it was when the nightmare finally ended. Yeah. And we stepped inside his room. He looked at me and said, "You guys want to know what he has?" And he told us that I had progeria and that the treatments was just here. So then, I went here in 2 years to do the week tests, and now I live here. I'm making, think, 3 years here. Yeah, maybe 2 to 3. And it's my birthday month. I'm going to be 13. Teenager. Which is a little scary word for me. Because, for me, I was 5 yesterday. But growing up is scary, guys. And day 24. Yeah, 24 in November. It's getting close. And now I forgot what it was. Let me think. Okay, now I live here. And it was just a lot different. I need to learn a new language and everything. Went to school. The first days, I hated it because I didn't understand nothing. And I kind of like school. Yeah, a lot of kids don't say that. But I do. And now I know English, and it's much better to speak with people, communicate, talk in school, understand things. And one of the things that I best like is to coming here and living here is to meet other kids with progeria. Because now I know 3 of them. Megan. Oh my God, I'm so nervous. It's like hard inside my head. And Sammy, and another girl from Brazil that she came here to do the treatments for the first time, and my mom helped her, her mother, and everything. And I forgot her name. I have very bad memory. But I was so happy to meet other kids because in Brazil I felt lonely. And my parents just put me in private school because they feared that I would get bullied in Brazil in the public schools. So in the private schools, I didn't. I had a lot of friends. I have friends and everything. But yeah, and this coming here and now because in Brazil, it's a very good place, but people are curious. So one time people stopped and asked me, what does he have? What is his disease? And my mom got very mad at that because she feared that would make me sad or something. And then here, that doesn't happen. I will say that you guys are awesome. And privacy, awesome. And yeah, it's just very good here. I love it here. And yes, I like my country. It's a very good place. But I like it here too. It's not like it was a big shock on me, but in Brazil, I move it a lot too. Because first I lived in a little farm, then after a city, small city, then a big city. So yeah, I'm very usual to changing places. And just waiting here, meeting other kids with progeria. And now I want to say that my speech is over. Thank you.
Carlos, you are a superstar. He told me I'm so shy. And he is not. I prepared a lot of questions for you, but you did everything alone. Great.
Thank you.
Great. I'm so happy to see such a young boy, a new teenager coming, and good job. So, you hear about 3 patients' stories. Sorry, my English is not is not good as Carlos and Megan one, of course. And but we have other families connected, right? Because they couldn't be here, but they didn't want to miss this beautiful workshop. They didn't want to miss you all, so they are connected with us. Here they are. Hello.
It's Enzo, Sammy.
Enzo, yeah. Can you hear us? They are from Australia, right? Hello. How are you doing?
Good. Hello, my name is Enzo. I'm from Australia.
And how old are you, Enzo?
11.
Okay. And can you present us your dad and mom?
Yeah. Hello, I'm Katerina.
Hello, I am Maxi. Hello, how are you?
Fine. We are fine. very happy to have you connected. So,
So happy to see you all.
Enzo, would you like to tell us something about your life? For example, are you going at school?
So, what I like to do at school is math, science, STEM, and art.
Oh, wow. Great subjects. And you are very active on the social networks. You are leading with your family the team Enzo Progeria, right? Can you explain us what this great project is?
Well, what we do here in Australia is trying to create awareness because most of the people haven't heard about progeria.
Yeah, because I'm the only one in Australia.
So you are unique.
Yes, I am unique.
Yeah. And what is the best part of this project? What do you like more of this?
Thank you for looking after us and helping us find a cure for progeria. You are amazing.
Great. Great answer. Can I ask you, just in confidence, we are just 150 people, so a great confidence. What's your secret dream?
My secret dream?
Yeah. Do you have one?
Is having a dog.
Oh.
I don't have a house yet.
Okay. Great. You will sure have it. So, and there are other families connected. So, Enzo, thank you so much. But remain with us for a while. I will switch to another family for knowing something regarding them.
Okay. Thank you for coming.
Thank you for having me.
Bye. He's amazing. I think we have other families right now. Who's the next? Oh, hello.
Hello. Hi, can you hear us?
Yes, we can. Can you hear us too?
Yes.
Okay, great. How are you?
We're good. Doing good.
And where do you come from?
So this is Clay. He's 4. We live in Central Illinois.
Oh, okay, so you're here from the States.
Yeah.
Okay. And it is a pleasure to meet you in virtually. And I would like to ask you, what's the most beautiful thing about being part of the progeria community? I know, such a great question.
Well, we appreciate that there is a progeria community and somewhere to go for answers. We're thankful for all they've done, and it helps our doctors a lot because most of our doctors have never seen a child with progeria. So there's somewhere for them to go and ask questions, and then they can help us better, and we are very appreciative of that.
And so Clay is 4, right?
Yeah.
And is he going to school?
So he goes to an early childhood classroom, which is kind of like preschool, sort of. Yeah, he goes So he goes to school 4 days a week.
Oh, great. Hey, good boy.
He's never done this before, so.
Oh, okay. So this is the first time. The first time for me to moderate a session, so not the first time this this meeting. Great. And is there something you would like to share with all this audience? I mean, we are like a great family. Something you would like to say them?
I don't know. I guess I'd just say, he's 4.5, but he's talking a lot more now. He actually ended up having a stroke in the womb. So he has hemiplegia. And so he had seizures for about 1.5 years, lost all of his words. And then now they've been gone for almost 1 year, and they're coming back, and he's doing good at school. And so we've got a lot to be thankful for.
Oh, okay.
And if he's with you, he would give you a fist bump, or no?
Yeah.
No. He really likes that.
Great. It's so beautiful. I would argue all, but we cannot because we are virtually connected. So thank you so much. I think it was very important for us to know you. And we have another family connected. Oh, I know them. They're from my country. Ciao. So, I know who you are, but what's your name? And what do you come? Okay, I can translate for them. He is Alessandro, he is 11, and he lives in Italy. So are you going to school, Alessandro? What's your favorite subjects? Qual è la tua materia preferita?
La mia materia preferita è matematica, perché mi vengono bene i conti.
Okay, his favorite subject is math. So maybe we have another scientist future. And Alessandro, what's your favorite thing to do with your friends? La cosa preferita da fare con gli amici?
Uscire al parco a giocare.
Okay, and cosa giochi? Okay. Okay. His favorite things to do with friends is to go out at the park and to play football. You know, we are Italian, we love football, soccer. And, okay, do you have a secret dream? Or what would you want to be when you grow up? Hai un sogno segreto? Cosa ti piacerebbe fare da grande?
Non lo so.
Okay. He doesn't know. You have time. You are so young. And for the parents, is there something? Do you have something you want to share with us, about the PRF community, or to say to this great audience hearing us? C'è qualcosa che volete condividere con noi della comunità progerica, o qualcosa che volete dire alle persone che sono qui?
Ciao a tutti, io sono Luca e fa piacere condividere il fatto che fin dall'inizio, quando abbiamo appunto iniziato il contatto con la PRF, abbiamo sentito di avere un trattamento familiare, qualcosa che andava al di là del rapporto medico, e quindi questo è stato e continua ad essere una cosa importantissima per noi.
Okay. He said, I'm Luca, and since when I started to know PRF, to come here in Boston for the treatment, the most beautiful thing was that there was not only a relationship doctor-patient, but there was a relationship that is the one we can feel in a family. So that is the most important thing, because we feel accepted, we feel comfortable, and we feel good. Great. And qualcos'altro che volete aggiungere? Is there something else you would like to say? What time is in Italy now? Oh, okay, 10 p.m. So tomorrow it is school time. Thank you. Grazie mille, thank you. Okay, so I think we are at the end of the session.
Great job.
I would like to do something. Can I? Okay. This is something of so American thing, an American thing to do. Because I'm Italian, and it is something different for me. And when I came in Chicago some years ago, I went to the mass, and there was a pastor teaching something about faith, and the audience was used to say, "Yeah, hey, come on!" So we are here today. It is such a very beautiful workshop. I cannot wait for hear what you have to say us in the next days, and to know everything about your research, your studies, and so on. But we are now enjoying our evening, our night. So what if you do the same? The best roar we can say all together. Is it okay? Okay. Would you like to do the countdown?
Okay. 1. Oh.
Yeah?
1, 2, 3.
Thank you.
Thank you.
Awesome job.
Whoa. I don't think I can follow that. Oh, I took the clicker and I didn't bring it back. Hold on. Thank you. I just want to let you know about my outfit. I wore this to feel, I don't know, good. It was brought here as a gift from a patient family from Kazakhstan. And oops, sorry. They said they wanted to bring some of their culture along so I could experience their culture like they were coming in for the trials to experience ours. So it means a lot to me. Whew, amazing. Thank you so much. You guys were spectacular, and that just made my year. So I'm going to give you a little bit of a chat for a little while and talk about lonafarnib journey to approval and lessons learned for future treatment and the cure. So just to give you a little bit of my experience, and then we'll have a wonderful singing session, and then we're going to take a group photo because we got to get all of this. This is an amazing group, and then have dinner. This is our first workshop since the FDA approval for the treatment of progeria. And I'm excited, we all are, to be celebrating that historic milestone with everyone here. And I'm going to tell you a little bit more about that journey. Our first drug approval is a rarity among rarities. In summary, we achieved a drug approval in an ultra-rare disease, very unusual, using investigator-initiated trials, also very unusual. And the approval was based in large part on data from a rare disease patient registry, which is pretty much unheard of. I think an important component to our first drug approval is that it gives us real inspiration for the promise of even better treatments and ultimately the cure for children and now many more young adults with progeria. The approval rests on the foundation of basic science performed by a small and committed group of progeria scientists whose ideas and hard work make a real difference. And that's vital, because we can only achieve better treatments and the cure together by standing on the shoulders of progeria researchers, many of whom, I'm going to say most of whom, are here tonight. So for the remainder of my remarks this evening, I'll highlight our extraordinary journey to lonafarnib drug approval, inserting some of the critical scientific milestones for those in the room who are here for the first time, and focusing specifically on the lessons learned to inform future treatment pathways and ultimately the cure. The idea behind lonafarnib for progeria came from a series of discoveries and experiments published between 2003 and 2006, starting with the gene finding. Thank you very much, Francis. Thank you very much, Maria, and all the other people who did amazing work, Tom and Ted and Mike Erdos and others, using cells from PRF cell and tissue bank in part, which I'm really proud of. And that showed us that the culprit was a single base pair mutation in the lamin A gene that yields the shortened and toxic protein progerin. So let me see if I can make this work. Oh my, I'm looking sideways. So basically, what you're seeing here is the C, the single base, just a C to T mutation. This is code sequence in lamin A, and this is progeria. And that just one needle in a haystack, that one letter, gives us a silent mutation, but optimized a cryptic splice site, or a splice site in this gene for lamin A that created a shorter protein, 50 amino acids shorter. And that has made all the difference. That's progerin, and that gives us progeria. Here I'm going to show you the post-translational processing of lamin A and of progerin. So on your left is lamin A, on your right is progerin. Both And, you know, it's really interesting because when we first discovered the gene mutation, this very small, powerful group of scientists who had studied this molecule, lamin A, sort of came into the fold, and that was really important. Both of these lamin A uses a side group called a farnesyl group, and that's shown here, oh boy, shown around here, to grab onto cell nuclei and essentially destroy cells. And there was a class of drug called farnesyl transferase inhibitors, or FTIs, as you see there on the right, that prevent the side group from being attached. And these are the drugs we explored in the laboratory for progeria. Proof of principle for our first drug trial was achieved by studies exposing progeria cell cultures and the brand new progeria mouse models, which could only be made because we knew the gene mutation, to FTIs. What you're seeing here on your left and the top is a normal cell nucleus, nice and smooth and round. Then you go down, you look at progeria, very abnormal, evaginated, kind of sick. And then treatment with an FTI, normalizing that cell culture. In the middle on the top is actually a slice from a mouse blood vessel, an aorta, major vessel. Nice and green and healthy with little blue, purple nuclei in there. And in the middle, progeria. Very unhealthy, very empty, very sick. And treatment with FTI, making that mouse aorta much healthier. And on the right, progeria mouse with and without treatment, looking much healthier. And because one of these drugs, lonafarnib, was already being used in clinical trials for children with cancers, we had the opportunity to do what's called drug repurposing, where a drug intended for one condition is applied to another condition much more quickly than if we had to develop a drug from scratch. So we formed a trial team at Boston Children's Hospital led by Mark Kieran and Monica Kleinman and me, and conducted what's called investigator-initiated trials, which is a research conducted by investigators who are not from drug companies. Now, it's important to note that these types of trials are not prospectively evaluated for drug approval by the FDA. We took this approach because at the time we knew too little about the natural history of disease in progeria to be able to present a suitable outcome measure for drug approval to the FDA. And we felt so strongly about getting a potential treatment to these children as quickly as possible, despite this limitation, because there wasn't a treatment and progeria is fatal, and we had to take action. And the FDA and our institutional review board agreed. And so in 2007, at the top, we started our first clinical trial for children with progeria, followed by a series of sequential trials, all single site and all open label, so that all of the children in the trial would receive drug. Our very first trial is here at the top, and we dubbed it PROLON 1, Progeria, lonafarnib, trial 1. And then we tested 2 more drugs after that in sequence on top of lonafarnib, and we called it the triple trial, which ultimately didn't show added benefit on top of lonafarnib as a monotherapy. And then on to a lonafarnib monotherapy extension with an arm called PROLON 2. Because we were building a program that not only tested new medications for progeria, but simultaneously, we were building a deep understanding of the natural history of disease because we felt it was the only way to truly discover ways to evaluate treatments in the future with the aim of ultimately having drugs that we could seek drug FDA approval for. So doing both the natural history and these drug trials simultaneously. Well, fast forward a bit to 2015, when Monica Kleinman and our cardiovascular expert from Brigham and Women's Hospital, Marie Gerhard Herman, and I visited the FDA in person, partly to talk to them about a cardiovascular measure called pulse wave velocity. This is a measure of blood vessel stiffness and consequently of strain on the heart. And throughout the trials, we had seen pulse wave velocity as a telltale sign of cardiovascular status in the children. It's abnormal early in life and progresses as the children with progeria get older. And we had seen it improve with lonafarnib therapy. So you're seeing pulse wave velocity extent on the y-axis, pre-therapy and end of therapy, and that bar across the bottom is normal. And so we were seeing improvement with lonafarnib. And so we took this to the FDA, and they said, while this was exciting, their response was that we had more work to do. Although there was support for pulse wave velocity as important for adults with heart disease, where it's actually predictive of the risk of heart attack in older adults, we had not proven that this measure was relevant to clinical disease status in the children. And you're going to hear more about that from our experts tomorrow and the next day, over the next couple of days. But during that meeting, we also showed them this slide. This was something we'd been working on for several years, but never considered as a possible approval pathway because lifespan actually wasn't a part of the clinical trial design. We showed them this initial survival comparison of children inside and outside of the trials. So what you're seeing here is survival on the y-axis, time on the x-axis. The bottom line is each time you take a step down, a child passes away. The bottom line comes from the International Progeria Registry, so outside of the clinical trials. And the top line comes from the clinical trials. And we combined the data from the Prolon 1, the first trial, remember this was only 2015, and the triple therapy trial. So this is all of that lumped together in comparison to these children that we had been tracking in the International Progeria Registry. And the FDA was intrigued by the survival data and told us that this might be considered if we were able to show clear and convincing evidence that lonafarnib monotherapy influenced lifespan. That one interaction sent us off on a multi-year project to gather as much information as possible and return with more data, if in fact we could show lifespan improvement using only trial data where children were only on the monotherapy lonafarnib. And we did. And we published this in the Journal of the American Medical Association in 2018. So what you're seeing here is just Prolon 1 and Prolon 1 plus Prolon 2. Again, the blue lines on the bottom are the children from the international registry, and the top lines come from the clinical trials, the red lines. So it was pretty exciting, and we thought we had something really important here for the FDA. And around that time, we initiated a partnership with Eiger Biopharmaceuticals to work in concert towards them submitting for drug approval and subsequently commercialization by Eiger. Now, during the application submission, we actually needed to update the survival data 3 times for the FDA. First, because the JAMA article, this article, was over a year old by the time we approached them for the first time. And then twice more because the process took so long to implement. Remember, there was 13 years of trial data. And when you submit for a drug approval, we couldn't just submit just the survival data. We had to submit head to toe everything that we had done. Remember, I told you we were looking at natural history. I mean everything. So there was this gargantuan data set that we had to organize and submit. Now, each survival update required new contact with everybody, our previously known and new children, and hard copy proof of patient status. And PRF could only achieve this because we're in contact with families as a community, keeping them informed about what was happening with the research and the trials. And it is our most important relationship, not only for situations like this, but also it keeps us squarely focused on our mission. I mean, do I even have to say that after what we just all saw? It's just spectacular. And all of this activity culminated in FDA approval for lonafarnib for progeria. We are joined by less than 5% of all rare diseases having any drug approved by the FDA. And I do hope in the future that changes dramatically. From there, European approval required yet another survival status update, as well as submission of some brand new data, new basic science, and data on progerin levels that wasn't available first time around that we're going to show you more of tomorrow. By the time, so when we submitted for the FDA approval, the label says we calculated a 2.5-year average lifespan extension. By the time the European approval came around, 4.3-year lifespan extension, which is a 30% increase in lifespan. Amazing. I know it. These updates and discoveries were only possible because the children were on drug without breaks through the serial trials and their monotherapy extensions. This is an almost unheard of strategy that we used. And I have to say, we just did what was necessary for the kids. It was just, what do the kids need? How can we do best for them? How can we help? What is the research that we need to do? So it's very unusual, 13 years. But for an ultra-rare disease, you have to be creative. And for our ultra-rare disease, this is what led to success. So, where do we go from here? I think this is a pivotal time for us all. We can evolve in ways that weren't available to us previously because, for example, we've done more work on outcome measures through all those natural history studies. And that work will be shared during the next 2 days. But we also face new challenges and opportunities. For example, how will we bring new discoveries made by the basic scientists to the bedside? How can we explore the pathobiology of progeria in cells and animal models and humans to find common disease features that allow us to move from bench to bedside with confidence? Will we be able to run shorter trials using proven biomarkers and clinical measures that we can evaluate for drug approvals over and above lonafarnib? This would help us, I think, to avoid unnecessary toxicities for drugs that may not help the children, they're just on for less amount of time, and make way for conducting multiple trials at once or in quick succession, which is pretty hard with an ultra-rare disease, but I think it's possible. We conducted open label studies because we felt strongly that leaving a child without any therapy was unethical. Couldn't do it. Now, will our trials be able to have a control arm with lonafarnib because the children are getting something that's helpful to them? Do we have common, reliable, and valid technologies available that will enable us to do maybe multi-site trials where the kids don't have to fly in from everywhere in the world to one place? And the trials we've implemented to date have all used repurposed drugs far into the drug development pathway. Can we take brand new molecules all the way from the culture dish to the bedside? And that's some of what we're going to talk about over the next 2 days as well. We're going to be seeing a lot of new science and spending a lot of time together. To me, this is the key to our future success, and it's why I'm so happy we're finally here in person. Sharing information and ideas and effectively communicating and linking between basic and clinical science. To me, it's the only way. If we can understand the pathobiology of disease in the human deeply and mechanistically through continuous connections between studies in the laboratory and in the clinical setting, our chances of discovering new strategies for effective treatment go up exponentially. So thank you all for being here to do this great work. And I think do you need this, Francis?
No, I'm going to have another.
Okay.
Could I get the microphone?
Oh my God, thank you.
I will be.
Good afternoon. Originally, I was supposed to follow that amazing panel that Sammy led, and I thought that was going to be really impossible. I made Leslie instead. But now I have to follow Leslie, so I guess it didn't turn out all that great for the order of events anyway.
Okay,
I'm Francis
if I get up and
Collins.
announce it.
I'm currently a senior investigator in the Genome Institute at NIH, but I'm also undergoing a detail to the White House, so I have to disclose that I'm also the special projects advisor to the President of the United States. But not right now. Right now I'm here because I care deeply, as you all do, about this disorder called progeria, more precisely Hutchinson-Gilford progeria syndrome, which has been an activity my lab has been engaged in now for more than 20 years. And the progress, as you have heard, and you saw that curve just now about the way in which publications are going like this, and they're not just publications that don't mean anything, they're publications that are leading us progressively towards advances. And it was just absolutely inspiring, Sammy, to have you lead our panel, because we haven't had the chance to do that before at one of these meetings. And if you wanted to be reminded why we're all here, that was the reason. So what a great way to kick off our time here together. I think, is Maria Erickson here? Has she arrived yet? Somewhere here. There. Maria, I think it was just about exactly 20 years ago, on a Saturday, working in my lab, trying to find the cause of progeria, that you called up and said, I've got this funny finding in the sequencing of this gene on chromosome 1, but it doesn't make a lot of sense because it looks like a lot of the kids with progeria, and we got those cells from Leslie and the foundation, a lot of them have the very same change in the sequence. It's a C that's mutated to a T, and that seems really significant. But then when I went and looked at the genetic code to try to see, well, what would that do to the protein? It doesn't look like it would have an effect. It looks like a silent mutation. It goes from GCC to GCT. That's still glycine. So, that must not be it. It's like, well, wait a minute. It only took a couple hours, I think, staring at the sequence to realize, oh, it's maybe silent at the protein level, but it's not silent in terms of its effect on splicing. And Leslie showed you that picture, and then we figured out, oh, that's it. This is creating a protein that's missing 50 amino acids. And as Leslie said, we were fortunate then. We landed in the middle of an area of really beautiful biochemistry and cell biology of people who had worked on this lamin A gene for years and never knowing that it would turn out to be so relevant all of a sudden to try to understand why this single letter change would cause this condition called progeria. And my goodness, then we all got to know each other really well. And the foundation made that possible with all the gatherings that were supported. And along the way, we understood rather quickly what the consequence was in terms of what this did to the health of a cell and to the nucleus of that cell and its shape. And you saw a picture of that. And it is rather amazing because that led within, I would say, weeks to the idea of a farnesyltransferase inhibitor as a possible therapeutic. And that timeline went by pretty quickly, but it was 4 years from gene discovery to starting a trial. That's almost unheard of for any disease, and certainly for a rare disease. And that just is an indication of how many people put their hearts and minds into trying to figure out how to quickly take that basic science discovery and move it forward. And that's what PRF has always been all about. Fund the basic science, figure out some of these really critical issues about how this particular protein does what it does. But don't stop there. Figure out how to turn that into some really interesting ideas about treatment. And now here we are at a very interesting place. And this whole meeting is just shot through with all kinds of cutting-edge research that has consequences for our understanding. And even, as you'll hear tomorrow morning in this session that I get to be the moderator, perhaps impending opportunities for clinical trials based on totally different approaches than a farnesyltransferase inhibitor, but maybe very complementary, maybe using RNA, maybe gene editing to actually go and fix that T that should have been a C using a gene editor. And you'll hear an amazing presentation tomorrow morning from David Liu, who is the guru of all of this. David Liu could work on almost any disease if he decided he wanted to. He is completely immersed in progeria because of his commitment to why this is so important. So it's pretty amazing to be here. And I too kind of am really glad to be together face to face after so many Zoom calls. I do apologize. I've got a cold, so I'm not going to hug anybody, and it's really killing me not to, because I don't want to give you whatever I've got. It's not. I want to reassure you, I am antigen negative and RNA PCR negative. I do not have that one. It's just a cold. You know, we can still have those things, I guess. We kind of forgot about that. But anyway, it's an inspiring time. And I guess 4 years ago, somewhat in an impromptu way, we ended up having a little singing as part of this. And Leslie, never missing a beat, decided, well, we should do that again. And I seem to have been asked to be the one to help you with that by perhaps leading a little bit of this. And again, because of this cold, I might sound a bit more like Rod Stewart today as opposed to James Taylor. But sort of trying to figure out what to sing at a time like this. Well, it seemed like we are all gathered here because we care deeply about this condition, which needs a lot of attention and a lot of advances. And as brave and bold as our progeria panel represented their situation, they need us all as friends. And we are friends. This is what PRF is all about. And we need to keep that forward and remember that we are there in whatever way we can be as scientists, as physicians, as just people who care. So maybe a song like You've Got a Friend doesn't seem totally out of place. And if you know the chorus, and I think a lot of people do, I hope you'll join in with me. When you're down and troubled and you need some love and care and nothing is going right. Have you had even your darkest night. You just call out my name, and you know wherever I am, I'll come running to see you again. Winter, spring, summer, or fall, all you gotta do is call, and I'll be there, yeah, yeah, yeah. You got a friend. If the sky above you should grow dark and full of clouds, and that old north wind should begin to blow, keep your head together and call my name out low. Soon I'll come knocking upon your door. Come on, y'all. You just call out my name, and you know where I am. I'll come running to see you again. Winter, spring, summer, or fall, all you gotta do is call, and I'll be there, yeah, yeah, yeah. So ain't it good to know that you've got a friend? People can be so cold, they'll hurt you and desert you. They'll take your soul if you let them. Ah, now, but don't you let them. You just call out my name, and you know wherever I am, I'll come running to see you again. Winter, spring, summer, or fall, all you gotta do is call, and I'll be there, yeah, yeah, yeah. You've got a friend. You've got a friend. You've got a friend. You've got a friend. I saw a few mouths moving, but they weren't very vigorous. Can now work a little bit into this here. 4 years ago, I looked back to see what was it we sang together, and it was this song, which I didn't have a cold time, so this is going to stretch my vocal cords a little bit. But it also has a chorus, and it's so appropriate when somebody says to you, well, you know, these rare diseases, they don't affect that many people, and, you know, it's really going to be hard to find answers for them, so maybe you should work on something else. Or maybe it's just not the time. Maybe it's too hard. We need to come up with brand new ideas that we don't have yet, so, you know, go back and work on something else, and maybe some technology will get invented. Well, sorry, that's not the right answer, is it? If somebody says it's not the time to find a cure for progeria, if not now, tell me when. That's the title of this song. And the chorus, all you got to know is, if not now, tell me when. There's one other line in there, I'll sing that. But if you can just do that part, no need to memorize anything, we can do this together. It's a song by Carrie Moonlighter. That isn't quite right, but something close to that. And I rewrote the words a little bit. And here's the chorus. Tell me when, if not now. Oh, tell me when. Moments or in little microphone problem. And we got a battery issue. Just cutting it. There we go. All right. When the microphone doesn't work, you say, if not now, tell me when. And they come right to you. Let's try that chorus again. If not now, tell me when. If not now, tell me when. We may never see this moment or place in time again. If not now, tell me when. Okay, come on, y'all, let's hear it. If not now, tell me when. That's better. If not now, tell me when. We may never see this moment or place in time again. If not now, tell me when. I see courage and vision in this place. This is for you. I see courage and vision in this place. And throughout all these struggles, you've modeled grit and grace. If not now, tell me when. If not now, tell me when. If not now, tell me when. We may never see this moment, place in time again. If not now, tell me when. So we'll work until it's done. Every daughter, every son, every soul that ever longed for something better, something brighter. All together now, there's nothing we can do. I'm talking to you. All together now, there's nothing we can do. Though sometimes we grow weary, love and hope will see us through. If not now, tell me when. Chorus, come on. If not now, tell me when. If not now, tell me when. We may never see this moment or place in time again. If not now, tell me when. One more time. If not now, tell me when. If not now, tell me when. We may never see this moment or place in time again. If not now, tell me when. If not now, tell me when. If not now, tell me when. All right, that sounded great, you all. Okay, one more, and then we're going to have a dinner or something, I think. Oh, don't forget the group photo. Okay, lock the doors so that nobody can leave. I hope you locked them already, or half the people in the back would be gone by now, but apparently it worked. This is a group participatory song that I kind of made up the words to while I was listening, but it's a camp song, and I saw that Progeria kids go to camp and have a pretty good time, apparently. That sort of tipped me off. Okay. So a song that you could just write new words to, where you only needed 6 words to make a whole verse. That sounds like the kind of thing that would fit nicely at a time like this. So the basic of the song, you'll probably know. And we'll sing the first verse in the traditional way. And then all the rest will be brand new for the first time on this stage. Oh, when the saints go marching in. Oh, when the saints go marching in. Well, I wanna be in that number. When the saints go marching in. Okay, that's the traditional one. And here we go. Oh, when we meet here face to face. Oh, when we meet here face to face. when we cheer for PRF. Oh, when we cheer for PRF. Well, I wanna be in that number when we cheer for PRF. Let's do that together. Oh, when we cheer for PRF. Yeah. Oh, when we cheer for PRF. Well, I wanna be in that number when we cheer for PRF. There's going to be clinical trials coming. You hear about things on the sort of drawing board with maybe Europe or something like that. That's always a moment when the trial starts to roll. Oh, when the trial starts to enroll. Oh, when the trial starts to enroll. Well, I want you to be in that number. Come on! When the trial starts to enroll. And this is a place, of course, it's about science, it's about hope. Oh, when the hope and love shines through. Oh, when the hope and love shines through. Well, I wanna be in that number when the hope and love shines through. Sing that with me. Oh, when the hope and love shines through. Oh, when the hope and love shines through. Well, I wanna be in that number when the hope and love shines through. Well, one more, and it's gotta be this one. Oh, when the race leads to a cure. Oh, when the race leads to a cure. Well, I wanna be in that number when the race leads to a cure. Do that again. Oh, when the race leads to a cure, leads to a cure. Oh, when the race leads to a cure. Well, I wanna be in that number when the race leads to a cure. Let's find that cure. Then we can really sing about it. And as Sam Burns would say, never miss a good party.